blessed, my Lord, I can see you in all the lovely things so fine and true. I see you in the beauty of the flowers and the rain. I see you between the lines of a sweet refrain. I'm so blessed, my Lord, I can see you. Even when I'm lonely and in pain, I see you in the beauty of the stars at night. I see you in my life, I feel alright. I see you, Lord, in sorrow and in happiness. I see you in the glory of sweet success. I see you, Lord, every hour, every day. I can see you, Lord, whenever I pray. I'm so blessed, my Lord, I can see you in the smile of a baby, sweet and true. I can see you in the eyes of my very best friends. I see you in these bonds, just like your love that never ends. I see you, Lord, in sorrow and in happiness. I see you in the glory of sweet success. I see you, Lord, every hour, every day. I can see you, Lord, whenever I pray. I see you, Lord. I see you with the glory of sweet success. I see you, Lord, every hour, every day. I can see you, Lord, whenever I pray. I pray. I can see you, Lord, whenever I pray. I see you, Lord.
Bayang magiliw, pelos ng silanganan, alam ng puso, sa dibdib mo'y buhay. Lupang hinirang, buyan ka ng magiting, sa manlulupi, kita masisingil, sa dagat at bundok, sa simoy at sa langit mo baka, ay nilang ang tula at awit sa paglayang tinamahal. Ang kisap ng watawat mo'y nagumpay na nagiging, ang bituin ng araw niya kailan ang magdidilim. Lupa ng araw na maraming pagsita, kulay langit sa likod. Aming ligaya na may mamagin, at namatay ng dahil sa'yo.
Hello everyone and welcome again to the second session of the PSAU Hainayan webinar series, a collaboration between the College of Arts and Sciences, Department of Biology, External and International Affairs Office, Innovation Center for Agroecology, Climate Change and Agripreneurship, and the PSAU Biology Society, organized by PSAU Live and PSAU Hainayan of the Pampanga State Agricultural University. We are currently on our third day, and we thank everyone here in the Zoom meeting, plus the viewers from our Facebook live stream for the support that you have given us on our past days. Once again, I am Jimwell Peñafiel, your host for today. Today's theme is Environmental Sampling Techniques, Soil, Air, and Water. Before I proceed, allow me to give a short recap on our past days. Last Monday, Sir J.J. Dida of the College of Forestry and Natural Resources of UPLB introduced us to the use of Kobo Toolbox and QGIS in environmental research, which can be applied on forest management and study site surveying. Yesterday, Sir Ace Amarga of Academia Sinica Taiwan and Ma'am Abby De Loyola of Herpwatch Pilipinas shared some of the methods and techniques that they employ in arthropod and vertebrate sampling and preservation. They even took the time to give a few inspiring messages to our audience. Okay, that's it for the recap. Moving on, let me acknowledge our Zoom participants and Facebook Live viewers. Please share our live video to support our today's session. Thank you. To maintain the order of this event, here are a few simple house rules to follow. Please make sure to mute your microphone while the session is ongoing. For our Zoom attendees, we would like to remind you to rename your Zoom display name starting from your institution's acronym and your full name. Also, may I just remind everyone that you may type in your questions for the speaker on our chat box for our Zoom attendees or in the comment section for our Facebook viewers. Lastly, the links for our attendance and post-evaluation will be posted in our Zoom chat box as well as in the comment section of our FB Live. Certificates will be given upon accomplishing the post-evaluation form, so please stay tuned and join us until the end of this program. For PSAU students, kindly use your personal Gmail accounts instead of your Eskwela accounts when filling up the form. With that, I guess all of us are now ready to meet our resource speakers for today. May I now call on Dr. Rafael R. Rafael to introduce our speaker.
Good morning, everyone. Our first speaker is Mr. Ram J. Dizon, which is a licensed environmental planner and a member of the Philippine Institute of Environmental Planners in Pampanga chapter. He finished his Bachelor's of Science in Biology, major in genetics at the University of the Philippines, Los Baños, and his master's degree in Environment and Natural Resource Management at the University of the Philippines, Open University. He is a senior environmental management specialist of DNR Central Luzon's Environmental Management Bureau. Our second resource speaker is Mr. Al Dimagibo, which is a Fishing Regulations Officers 1 in BIFAR Region 3. He finished his Bachelor's of Science in Biology and Master of Science in Biology at PSAU. He is currently the regional focal person of Balik Sigla sa Ilog at Lawa project, and he is also involved in the climate resilient tilapia farming in the Philippines and part of the collaboration project of DA-BIFAR and UNFAO. Hope everyone have a productive and active learning. Don't hesitate to ask questions to our resource speakers. Thank you and God bless.
Now we will proceed on our lecture. May I call on Sir Ram J. Dizon?
Hello. Hello, good morning, everyone.
Good morning, sir.
Okay, so my topic is environmental sampling techniques for water. So let me just share my screen first. Okay, so water sampling procedures for environment. Okay. So for water sampling, okay. So we will be discussing personal qualification and training, representative sample, steps in water quality monitoring plan, sampling methodology, chain of custody, and summary. Wait lang, sandali lang. So, okay. So just an overview sa work namin in environmental management bureau. We are tasked to monitor yung water quality in terms of ambient water quality, ambient water monitoring, and effluent water sampling. So in ambient water monitoring, we are checking and evaluating the physical, chemical, and bacteriological constituents of surface water bodies. And when we are talking about what effluent water sampling, it's a process of checking, evaluating, or investigating the quality of the effluent or wastewater discharges from industry and establishment or facility. Okay, so usually separate yung discussion with in terms of conducting sampling on ambient waters and effluent waters kasi iba yung parameters na ginagamit dyan. Pero halos similar rin naman yung mga process. So, pag na-confuse kayo kung pang ambient water or effluent water, just ask me. Okay? So, for personal qualification and training ng sa pag-conduct ng sampling, sa amin kasi rini-require before you conduct water sampling, kailangan nakapag-perform ka na ng necessary skills or you have undergone training sa calibration at saka pag-collect ng sample. Otherwise, you need supervision from those na meron ng mga may training na doon. So usually, for third parties or yung mga nagko-conduct lang ng research outside ng bureau namin, madalas nag-hire sila ng third-party accredited laboratories, DNR accredited laboratories to conduct yung mga sampling at saka analysis ng mga water samples. Okay, so we proceed regarding to sa sample. What is a representative sample? Okay, so it's a small amount of something that truly reflect the whole, and it must be accurate. So this is important, especially in environmental water sampling, kasi when we are identifying stations where we need to collect samples, we need to be strategic about it. Kailangan na nare-represent niya yung per area ng water body, in case of water body, or yung entire pollution source, wastewater pollution source ng isang establishment. So kailangan mapinpoint natin kung saan tayo dapat kukuha ng sample. At syempre, kailangan enough yung amount niya to represent yung data na kailangan natin. And then it must be accurate. Yun nga, kailangan yung tama yung lugar, and tama yung amount, and tama yung mga mula sa container hanggang sa pagkuha ng sample. Okay, so how can you prove that a sample is a representative? So since samples are evidence, especially sa bureau namin, kasi minsan nagagamit siya during hearing kapag mayroong mga violations or investigations. So nagagamit talaga siya as proof. So they need to be credible to court, public, and regulated community and government officials kapag pinipresent siya. So kailangan properly documented, pati yung procedures na susunod, and yun nga, nasusundan pati yung mga calibration, pati yung mga pagkuha ng sample, kailangan nasusunod lahat yung procedure na yun. Okay. So, mostly ng sampling natin, malaki talaga yung work prior sa conduct ng sample. So, in the steps in water quality monitoring, isang pinaka-importanting gagawin natin ay yung preparation of a monitoring plan. Either kung ambient water man yan or effluent ang conduct natin ng sampling, kailangan meron na planado na siya sa umpisa pa lang prior sa pagpunta natin doon sa site. Then we need to collect samples. Then yung field tests and measurements, if we need to conduct on-site testing. And then, siyempre, pag-record ng mga observation, on-site results, at saka field activities. And then, pre-treatment, preservation, and transportation of samples to ensure na hindi mako-contaminate yung sample. Then, laboratory analysis, data processing, interpretation, analysis, and storage, and then preparation of report. So, ang di-discuss natin hanggang doon sa pre-treatment kasi nga sampling techniques lang to, and yung iba yung mas mahaba yung discussion for laboratory analysis. So, pero dadaanan din natin ng konti. Okay, so ito nga, yung preparation of monitoring plan should be prepared ahead by the supervisor or the team leader. Okay, so what is this monitoring plan? So it is a report that describes how the water body will be monitored and how the water quality will be measured. Okay, so a well-designed monitoring plan will help ensure that the procedures for water sampling and other activities will conform to the objectives of the monitoring. So dito, it's important na meron ka monitoring plan kasi nga dyan nakalagay kung ano yung mga kailangan mong gawin. So hindi basta-basta ka lang pupunta sa site tapos kukuha ng sample. Sabi ko nga, it must be accurate. So mula sa pag-prepare ng mga gagamitin, sa pag-schedule ng mga calibration, at saka sa pakikipag-coordinate sa mga tao sa labas ay na taga doon. And also, yung mga pag-identify ng kung anong mga kailangan mong makuhang mga information dun sa lugar. So the components of a monitoring plan is background information, objective of monitoring, monitoring stations, water quality parameters for measurement, timing and frequency of monitoring, water quality sampling and test methods, quality assurance and quality control procedures, and budget for the monitoring. So kapag na-fill up mo yan, kung pampapansin nyo, halos kompleto na yung information nyo, except na lang for the sampling itself. So, and kung familiar kayo kasi mostly BS biology kayo, di ba? So mostly siguro may nagti-thesis kayo, nagkakonduct ng research. So halos kamukha siya ng thesis na ng mga title defense nyo, di ba? So conduct kayo ng background information and then yung objectives and then yung mga methodologies natin. So kailangan, alam na natin mga gagawin natin before pa tayo pumunta dun sa lugar. So in obtaining background information, okay, so you need to conduct preliminary survey if it's your first time na magko-conduct ng sampling doon sa lugar na yon. So, or first time mong pupunta doon sa water body na yon. But in kunyari, you are conducting a study na prior to that, meron na nag-conduct na study na yun, pwede mo nang obtain yung ginawa nilang survey dun sa lugar. But if it is your first time, you need to conduct a preliminary survey and there should be like a preliminary site assessment form na fill upan mo. So ito yung example ng site assessment form na ginagamit namin if we're going to conduct a preliminary survey. Pwede mo siyang gayahin, pwede rin naman gumawa kayo sarili nyo. So mostly information lang naman yan, yung stretch ng river, yung name ng river. Pag mag-identify kayo ng station, you can also name yung i-number nyo yung station na yun, kung first station nyo siya o hindi. And then, if you can fill up yung mga information like description ng flow, tapos kung pa-upstream siya or pa-downstream, kung and then yung dimensions ng river, yung width niya, tsaka yung depth. Usually, hindi lang kasi isa yung kapag minimeasure natin yung depth, minimeasure natin siya from yung dulo to dulo yung river para makita natin talaga yung kung gaano siya kalalim. And then yung velocity, if you have yung flow meter, you can check yung velocity nung flow ng river. And then, kung anong type ng nature niya, kung bedrock, sandy, clay, And then pati yung vegetation at saka yung classification, river classification yan. If you are not familiar with the river classification, actually sa Environmental Management Bureau din, kami rin yung nagka-classify kasi ng mga water bodies. You can go to our site, emb.gov.ph, and then go to the water section. At tapos meron sa page doon to check kung anong classification ng river water body. Tapos pero kung hindi siya classified, usually you can also identify kung ano saan siya ginagamit, either for recreation or for swimming or for agriculture. Tapos yun yung ilalagay nyo. It's important kasi especially if you are checking the quality of a water body, kasi naka-base sa classification niya or sa use niya kung ano yung mga guidelines for the parameters na para masabing maganda pa siya or hindi yung quality ng water body na yun. And then tapos we need also to determine kung saan siya malapit, kung sa mga residential area, industrial area, and then kung anong type of industries rin ng possible na maka-effect ng water quality niya. If there are quarrying, smelting, manufacturing, or processing. And then accessibility. It's important also na ma-identify natin kung accessible ba yung site na yon. Kasi isa sa mga priority natin to identify, to determine kung maganda siyang sampling station is kung accessible siya at safe siyang puntahan. Hindi natin pwedeng compromise yung safety natin when we are conducting sampling. And then yun nga, if we assess kung madali siyang puntahan at kung maganda siyang representative for that station, we can propose it as a sampling station. If not, kung may mga problema doon sa lugar na yun na para hindi ka makakuha ng sample, we can also choose not to propose it as a sampling station. Okay. And then in conducting preliminary surveys, we can choose three methods. One is field survey. And then, okay, sorry. We can choose one is secondary data collection and analysis, na kung saan from other studies, pwede tayong magkumuha ng information doon. And we can also gather information from like yung mga mapping, topographic maps, or ngayon, kahit Google Earth, we can determine para makita natin yung mga mga information regarding the sa river. Kunyari, kanina, di ba, kung anong mga nearby resident areas na nakasurround dun sa area, kung gaano siya kahaba. So, without going to the site itself, pwede na natin ma-determine yung mga information na yun through maps, related studies, at saka yung mga information na makikita natin publish online. And then, we can also do coordination with local government units and other government agencies na nagko-conduct na sample doon. So, yan. So, mostly kasi madalas naman, para hindi na natin pahirapan yung sarili natin, we conduct coordination na lang sa local government units or sa provincial environmental officers doon, para dun sa mga existing na mga information nila. Now, if kulang pa rin yung information na yun, then we can go ahead and conduct field survey. So, pupunta dun sa site, pupunta dun sa area to assess mismo, tayo mismo yung mag-assess ng area na yun. Okay. So, in field survey, okay, before ka pumunta sa isang lugar, so kailangan plan ahead na. So, kailangan meron ka ng contact. Mostly local government unit ang preferable na contact mo. And then verify the weather forecast. Especially kung magko-conduct ka na ng sampling, it's important to verify yung weather forecast. Kasi minsan kung malakas yung ulan, it could affect the quality of the water. So verify mo na yung weather forecast prior pa pumunta doon. Kung bumabagyo, I would suggest you reschedule na lang. And then, prepare a timetable. Kung first meeting pa lang, second day, pupunta ka sa lugar or makikipag-meeting sa community. So prepare a timetable kung ilang araw ka doon at saka kung gaano katagal yung papunta doon. Ensure provisions for logistics, especially kung gagamit ka, kailangan mo ng bangka, syempre, or mag-hire ka ba ng ibang sasakyan papunta doon. So ensure na prepared na yun, especially kung kailangan mo ng bayaran agad. And then prepare materials and equipments. Kung yung mga GPS maps, kung may GPS kung meron kayo, compass, maps, usually mga na-download na yan sa cellphone, so pwede na rin yun. Pero mas maganda kung may GPS kayo kasi there are areas, lalo na kapag remote areas na hindi connected na signal. So hindi rin maganda na dependent tayo sa mga cellphones natin. So maganda meron din kayong mga maps na printed, especially topographic maps. And then other information kung kailangan yung ma-check yung taas ng lugar. So you can use the altimeter or mga record books, safety aid kits then in case na medyo yun nga masukal yung lugar na yun, hindi mo iwasan baka magkaroon kayo ng sugat. Okay. And in field survey, we also conduct visual survey, participatory mapping, and interview survey. So with visual survey, ito yung usually kapag field survey naisip natin, we go to the site mismo, maglalakad tayo papunta sa site to confirm yung mga initial na findings natin or information na nakuha from other preliminary survey. So, one, ito yung mga pinaka-mainam kasi we can personally check kung ano yung mga tama ba yung mga information na na-plot natin, especially kung nakadepend lang tayo sa map. Kunyari, doon sa map pala may tulay pero kapunta natin doon sira pa yung tulay yung ginagawa. So, madidetermine natin yung talagang actual situation ng lugar na yun. And then we can confirm yung condition ng water din kasi hindi rin natin naman makita kung magbe-base lang tayo sa Google Earth for example, hindi natin makita talaga kung ano yung situation ng river mismo, yung conditions ng water body. And then we can also enable yung designation of monitoring stations. So kung once na napunta na tayo dun sa lugar, we can check kung talagang maganda siyang pagkuhanan ng sample. Okay, so we can fill up yung, for example, visual stream survey when we conduct visual survey. So ito gaya nung kanina na site assessment pero mas detailed lang siya. So i-determine mo kapag nandun ka na kasi, example lang to, you can choose to omit yung mga other information kasi masyado siyang detailed. So like yung mga kung anong type ng mga establishments na nandun sa floodplain, yung lugar, yung area na malapit dun sa river. So kung industrial ba siya, commercial, residential, and then yung riparian cover, yung banda dun sa side ng river, kung ano mga halaman nandun, tapos yung sa river banks, kung ano din mga nakatanim, ano mga nakalagay dun. And then other information like water flow, flow rate, tidal influence, bed composition. So mostly, hindi mo naman kailangan i-fill up lahat kung hindi mo ma-obtain yung information, but it is just a guide kung ano yung mga possible na gagamitin mong information dun. Depende sa research mo or sa study na gagawin mo dun sa lugar. So dadaan ako na lang to kasi medyo mahaba siya. So yun nga, it will depend on kung ano yung goal mo or objectives mo sa pag-conduct ng study, kung ano yung mga importanteng information na kukuhanin mo. So even yung mga aquatic biota, yung mga nakatira doon sa lugar, kung makikita mo sila, you can also fill that up. So yun nga, mahaba siya. So, actually makikita siya sa manual. Mamaya i-share ko yung link. Kung interested kayo gayahin siya, pwede din. So, aside from visual survey, you can also conduct participatory mapping. Actually, I would suggest you conduct participatory mapping prior sa visual survey. Ito yung using a map nung proposed area of study nyo. Magko-conduct kayo ng community meeting, community discussion with local residents at saka indigenous communities dun sa lugar. As information kung paano pumunta, kung safe ba doon, or kung ano pa yung mga possible na makikita yung mga conditions dun sa lugar na pupuntahan nyo. So ito yung example ng ginagawa namin usually through Google Earth bago kami pumunta doon. Simple lang naman, i-check, this is Umira River sa Aurora. And i-check mo yung lugar, tapos mag-identify ka ng mga possible monitoring station na pwedeng samplingan. Ang preferred namin usually mga may tulay, kagaya doon sa Station 1 dito. But there are cases na after nun wala nang mga accessible na tulay or accessible na daan. So yun yung mga possible na isipin namin na kakailangan namin magbangka. So prior pa that, mag-prepare na kami na makipag-coordinate at mag-hire ng bangka papunta dun sa mga water bodies na yun. Okay. Sorry, parang okay. And now for sa ano naman sa mga if you're going to conduct effluent sampling naman, syempre ang kukunin nating information ay information regarding sa facility. So we can gather this information through either coordinating with the facility, kasi syempre usually naman kung pumayag sila magkunda ka ng sampling sa lugar sa place nila, cooperative naman sila. But kung medyo mahirap sa kausap, you can go to the local government units at sa kanila rin kayo makipag-coordinate. So mostly ang kailangan natin, vicinity map ng lugar to identify nga kung saan tayo kukuha ng sample, including yung plant layout, yung manufacturing process flow, kasi dyan na ma-determine kung ano mga possible na mga parameters or pollution na dinidischarge nila, source ng water supply, wastewater generated from different processes, kung ano man type ng wastewater nila, treatment methodology nila, kung ano, paano nila dinilinis yung dinidischarge nila, yung nga, discharge points, sampling points, kung saan tayo pwede possible na kumuha ng sample, production level, number of discharging days, kung tuwing kailan sila nagdi-discharge. Kasi usually, hindi naman yan araw-araw nagdi-discharge. So, dapat natatimingan din kung saan kailan tayo kukuha ng sample. Volumetric flow rate of wastewater generated per outlet kung available lang. Kung saan sila nagdi-discharge na body of water, at saka kung ano yung mga waste minimization nila. So, you can coordinate with local government units or sa EMB, sa amin, sa Environmental Management Bureau to collect kung anong possible na information yung mabibigay namin. And then, we can also conduct interview survey. It's similar sa participatory mapping, but this one must dun sa mga credible person tayo makukuha ng information. So it's a group interview and it is recommended kasi nga may mga makakapag-produce siya ng mga readily results na kakailangan natin if we're going to conduct sample. So we need to be serious in identifying yung mga key informants sa interview survey. So, kailangan silang enough knowledge, ample knowledge dun sa area. And usually, ito yung mga local community officials like barangay officials, yung mga environmental officials ng mga lugar, tribal leaders, elders, at saka yung mga model citizen teachers, at saka yung mga community achievers. So, madalas yung mga nagko-conduct na study din, mga local na nagko-conduct na study din sa area, we can organize an interview survey with them para mas mapadali yung pag-gather natin ng information. So, sample ng isang questionnaire na pwede nating tanong during participatory mapping and also interview survey. Ito, yung mga name ng barangay, name ng mga ng river, at saka kung saan siya nag-originate. Yung usually, yung makikita natin doon sa visual site survey, pwede nating itanong natin sa kanila. So sample lang to, you can also modify yung mga tanong para depende nga dun sa objectives ng study nyo. Okay. So and then, okay. Sabi ko lang, so objectives naman. So it's important na clear tayo sa objectives natin sa monitoring. So kasi depende sa objectives natin, malalaman natin kung anong mga parameters yung kailangan nating kailangan nating i-test. And kapag alam natin yung mga parameters, alam na rin natin kung anong mga kung ilang containers yung dadalhin natin, kung anong mga preservation na kailangan natin gawin, anong mga preparation na kailangan natin gawin prior tayo sa pumunta sa site. So in ambient water monitoring, usually, mga common objectives yan, do we classify the water body, kung tama ba yung classification niya, trend monitoring, kung for example, sa isang year, yung quality ba niya naaangkop pa sa pinagagamitan ng water body na yon. So chine-check natin siya either every month or quarterly, depende sa objectives nga. And then designation of non-attainment area. Kung yung lugar na yon highly polluted na at hindi na dapat pang madagdagan yung pollution doon. So we can declare it as non-attainment area. Or sa amin, if that area is surrounded by industries na mga possible na makapollute sa water body, check namin yung ECC nila, yung Environmental Compliance Certificate nila, and check natin kung kasama doon yung minsan din-require natin sila na kasama sa nakasama sa mga obligation nila is to conduct ambient water monitoring dun sa mga water bodies na malapit sa kanila. And then we can also monitor kung may mga possible na mga water-related problems doon, or kung kailangan natin mag-conduct ng baseline data. Usually baseline data ginagawa yan prior sa conduct kunyari ng pagtayo ng isang project dun sa area. So meron tayong baseline data kung ano yung original composition at saka quality ng water body dun sa lugar prior na sa operation ng isang kunyari manufacturing plant. Or scientific studies din if we are going to do research, and then also for other purposes na hindi necessary. And then for effluent monitoring naman, we conduct effluent monitoring. Siyempre, one, if we want to check kung effective ba yung treatment na ginagawa ng isang company, so we conduct effluent monitoring and then identify potential environmental problems kung possible kapag nag-discharge yung nag-discharge sila ng wastewater. And then in case na sa permit nila, yun, nagko-conduct din ng effluent monitoring. kung meron silang discharge permit. And the detect and characterize report on releases by the facility establishment. Kung meron bigla sila nag-discharge dun sa lugar without any permit, pwede natin i-check kung maganda, kung polluted ba, pollutive ba yung wastewater nila. Determine yung impact niya sa receiving body. Kung nakita natin na, kunyari, nag-initial conduct tayo ng ambient water monitoring and nakita natin doon sa trend monitoring, papapangit yung water quality ng river, we can conduct effluent monitoring doon sa mga industries naman doon. Tsaka to check kung talagang sila yung may kasalanan kung bakit pumapangit yung quality ng water body na yun. And then we can also determine yung effectiveness ng waste minimization or kung ano mang technology yung ina-apply nila dun sa kanilang company. Okay, so in determining monitoring stations, yun nga nabanggit ko na, it's very important na we prioritize convenience, accessibility, and safety. Okay, and then we need to collect samples from well-mixed flow. So either ambient ba yan o effluent, we need to determine kung saan talagang nakamix yung mga lahat ng mga portion ng mga water body na kukuhanin natin. Okay, so in reverse, we need to collect samples at the center of the channel. Kasi yun nga, yun yung sure tayo na well-mixed yung water doon and ma-avoid pa natin yung disturbance dun sa mga riverbanks na possible na maka-affect, like for example dun sa mga kapag na-disturb natin yung banks, baka lumabo yung tubig. So mas maganda doon na tayo sa gitna ng channel kumuha. So usually naghahanap kami ng stations na may tulay nga kasi nga mas ma-accessible agad siya para kuhanan ng sample sa gitna. Collect sufficient volume of water samples. Doon sa monitoring stations na yun, kailangan makakakuha tayo. Enough siya para makakuha tayo ng kailangan nating volume. Okay. And in selecting monitoring stations sa mga rivers, ito yung mga suggested na mga qualification natin. Not necessarily na lahat to kailangan ma-check natin, ay kailangan na dun sa water body kasi that's impossible. But pwede nating i-consider yung mga to when we are determining yung mga monitoring stations. So, points immediately before the inflow of a river or stream into a marine water body. So, yung malapit sa river mouth, maganda meron tayong station doon. Siyempre, yun yung minsan nagmi-mix na yung tubig alat at tubig tabang. So, maganda na ma-check natin yung quality niya prior sa pagpunta niya sa isang marine water body. Points along the main river, downstream of confluence and upstream of the confluence with tributaries or drainage channels. So, usually naman yung mga rivers kasi meron sila mga tributaries. So, we can select sampling points or sampling stations prior dun sa confluence nila, pagsasama ng dalawang rivers and after nung confluence na yun. Pero may note dito, sites immediately upstream or downstream from the confluence may not immediately mix and can produce backflow that can upset the deposition of flow. So when we are targeting those stations na before and upstream and downstream ng confluence, mas maganda hindi dun sa exactly malapit mismo dun sa confluence. Kasi nga, possible na hindi pa naghahalo yung mga water nun from yung tributary at saka yung main river. So, mas maganda medyo a few meters after nung confluence, dun tayo kukuha na sample. At areas public use of water contact recreation like swimming water use. So, yung mga kung maraming pasyala, yung isang part ng water body na yun, pasyalan at nagsiswimming yung mga tao, we can choose to have a monitor station doon. Siyempre, especially kung gusto natin malaman kung appropriate siya for skin contact activities. Then points along the river where there is marked transition in topography such as waterfalls. Kung meron mga significant na mga change sa topography ng lugar, yun nga, gaya ng waterfalls or biglang elevated yung lugar, we can also choose to have sampling points doon. At habitat areas of sensitive species, syempre kung may mga endemic dun, lalo na kung may mga endemic na mga species doon sa lugar, we can also set monitoring station doon. Variability of flow patterns by artificial physical structures such as dams, weirs, and wing walls. So kung mayroong mga tinayo doon na nakakapag-obstruct ng mga water flow, not just dams, minsan pati mga bridge or temporary bridge nakakapag-obstruct ng water flow ng isang river. So we can set a monitoring station doon kasi minsan may effect din yun sa water quality niya. Kung tributaries naman yung kinukuha ng sample, same sa mga main rivers, minsan near sa mouth din, pwede tayong kumuha ng sample. And then when there are suspected point sources of pollution or non-point, point sources yung mga ma-identify natin yung mismo kung saan siya nagdi-discharge. So like wastewater treatment plants or non-point like yung mga feedlots, yung mga irrigation canals. So yung hindi natin ma-determine exact discharge niya, kung kukuha tayo ng sample sa river, kung maganda, mayroon tayong monitoring station malapit din sa mga areas na yun. For stream with less than 20 feet wide, sample should be selected where water is well mixed. So, meron kasing mga streams, usually yung mga hiwa-hiwalay yung flow ng river, so, and also mababaw masyado. So, mas maganda, yun tayo sa makakapag-collect tayo ng sample na well mixed yung water niya. Okay. So for monitoring of river, usually we need at least 3 sampling stations. So gaano man siya kahaba, at least 3 sampling station. Yung upstream, midstream, and downstream. Or kung mga 1 kilometer or kung mas mahaba yung river, pwede tayo determine a station like every 5 kilometer stretch of the river. Kung mahaba, like for example, Pampanga River. Pero pwede rin per partition, but at least minimum of 3 sampling stations. Or yun nga, bukod sa minimum of 3, pwede tayo nga mag-strategize na mag-determine ng mas maraming station na nagre-represent nga dun sa mga areas. For example, dito sa map, naglagay sila ng sampling station malapit dun sa may cattle farm, dun sa hydropower dam. So may before and after nun, meron silang sampling station. Tapos merong mangrove forest, so may malapit na sampling station dun sa mangrove forest. Tapos merong lake na malapit, so meron din station doon. Tapos may public bathing area. So may mga station din sila dun sa mga lugar na yun. So maganda yan, kaya nga kukuha tayo muna ng preliminary mga background information para ma-identify na natin kung anong mga stations na kukuha, kung saan tayo maglalagay ng mga sampling stations. So yun, samples may be taken at any accessible point representing each river section. And now for marine naman, monitoring stations, coastal and offshores. So usually for less than 1 kilometer beaches, gaya na river, usually 3 monitoring sites pwede na. For more than 1 kilometer stretch, we can have like 300 meters apart na monitoring stations. So, but we are collecting sample dun sa mga representative ng mga bathing area. So, usually, para masasabi natin bathing area, dapat saan madalas naliligo yung mga tao. So, ang reference namin dyan, yung chest level yung lalim ng tubig. So, kasi doon talaga madalas nagsuswimming yung mga tao. So, doon kami kukuha ng sample. Okay. Now, for effluent naman, so, effluent kasi dinidescribe siya ng Clean Water Act as discharges from known source which are passed into a body of water or land or wastewater flowing out of a manufacturing plant or industrial plant, including domestic, industrial, or recreational facilities. So, kukuha tayo ng sample or magsiset tayo ng monitor station kung saan yung wastewater nila papunta na sa isang receiving body of water. Not necessarily after ng isang wastewater treatment facility, pero kailangan papunta na siya sa isang body of water or land. Minsan kasi hindi naman body of water yung tinatapunan nila. So minsan sa lupa lang. So dun sa pa-discharge nila, dun na tayo kukuha ng water sample. So yun yung station natin. So it's important na meron tayo ng plant layout nila na information para ma-determine natin saan sila nagdi-discharge. Minsan hindi lang isa yun. So like dito sa map, nag-set tayo ng mga sampling location, yung color blue kasi marami silang discharge point. So, must be downstream of all treatment processes. So, yung papunta na nga dun sa after ng mga treatment nila, papunta na dun sa papatapon na dun sa water body or land. And then, flow rate can be measured or estimated din para malaman natin kung gaano kalakas yung discharge nila. Usually may information na yan kasi require yung nagdi-discharge na ma-determine yung flow rate nila. So you can ask yung flow rate nila ng mga company. And then yun nga, should be safe and accessible din. So kami, require namin na safe and accessible yung discharge point nila kasi nga minomonitor yan. Pati ng in-house monitor, pati sila nag-conduct ng self-monitoring. Okay. So sampling technique. Okay, so yun nga, we need to collect sample do sa final outfall. But there are cases kasi na hindi accessible yung final outfall nila. Now, kapag hindi sa accessible, doon tayo sa septic tank kukuha, malapit doon sa filter. Doon sa last chamber ng septic tank, prior dun sa effluent filter. So doon tayo kukuha ng sample for monitoring. Okay, so bago nga tayo mag-conduct ng sample, we need to determine kung anong mga parameters yung ko-collect natin. And for ambient water monitoring, depende yan sa objectives natin. But usually, we have yung primary parameters and secondary parameters. If we are checking yung trend, kunyari trend ng water body, or kung naaangkop pa siya for dun sa use niya or classification niya, we can choose to monitor yung mga primary parameters lang, like yung biochemical oxygen demand, chlorides, color, dissolved oxygen, fecal coliform, nitrates, phosphate, pH, temperature, at saka TSS or total suspended solids. Okay, but yung biochemical oxygen demand and chlorides, usually yan for sorry, mali yung nakasulat kasi. Usually yan is for inland waters sa mga freshwater bodies. Itong mga parameters na to, wala sila sa coastal and marine waters. Kasi yung chlorides, alam naman natin maalat yung tubig. So madalas mataas yung chloride content niya dahil sa sodium chloride at saka sa mga ibang salts na nandun sa water body na yun. So mataas na definitely yung chlorides niya kahit hindi pa dahil sa mga pollutants doon. And then biochemical oxygen demand naman, kapag mataas yung chlorides ng lugar, minsan nagkakaroon siya ng false results na nakakapag-effect doon sa BOD niya. So hindi na namin tinetest yung biochemical oxygen demand doon sa lugar. So yung mga parameters na to, hindi ko, since something techniques yung discussion natin, hindi ko na siya di-discuss in details, but there are parameters usually now what to determine nga yung water quality ng water body. So, especially yung biochemical oxygen demand, yan kasi yung oxygen na consume ng mga microbacteria at saka mga kapag nade-decompose yung mga matters dun sa water. So, maganda siyang indication kung maganda pa or pangit siya. Kung mataas yung BOD, usually, ibig sabihin, polluted yung water body na yun. And then secondary parameters. So kung kunyari iba yung objectives natin to determine, like for example, we need to determine kung yung effect ng mining sa sa water body na malapit sa kanila. Siyempre, hindi sapat yung primary parameters. We can also conduct sampling, like for example, for the heavy metals dun sa lugar na yun. So depende siya sa objectives natin. Okay, so ngayon naman, for effluent naman, effluent parameters, depende yan sa industries na industry sector na kukuhanan natin. So, kung magko-conduct kayo ng test for the effluents, we need to identify kung anong klaseng industry yung kukuhanan natin ng sampling. And then we can refer dun sa DNR Administrative Order number 8, series of 2016, o yung DAO 2016-08, yung Water Quality Guidelines and General Effluent Standards. At dun ka makikita mo kung anong type ng industry category. For example, dito, restaurants, food chains, bars, and food beverages, yung mga kailangang monitor na mga parameters dyan, yung BOD, yung biochemical oxygen demand, total suspended solids, oil and grease, at saka surfactants. At kung hospitals naman, ang kukuhanan natin or mga medical facilities, ito naman, color, temperature, pH, BOD, TSS, fecal coliform, ammonia, nitrate, phosphate, oil and grease, and surfactants. Kung palengke, mga public markets, similar, color, temperature, pH, BOD, total TSS, ammonia, nitrate, chlorides, oil and grease. So, depende yan sa industries. So, maganda may reference tayo, like ngayon, check natin yung sa DAO 2016-08. Okay. So, ngayon na we have yung mga information, doon na tayo sa sampling method. So, we have two types of sampling methods. One is grab sampling, and second is composite sampling. Grab sampling, yung pagkuha lang sa isang sample, a single water sample collected at one time from a single point. So, kuha ka lang ng sample sa isang lugar, minsanan, yun na yung ipapa-analyze mo. So, yun yung grab sampling. Composite sampling, as the PNSW of 2007 defines as composite sample as a series of individual grab samples taken at different times from the same sampling point and mixed together. So yung definition niya, parang series siya ng grab sample na kinukuha sa iba't ibang oras sa isang sampling point. But there are other types ng composite sampling. So how do we determine kung grab sampling kukunin natin or composite sampling? Kapag grab sampling, usually ito naman yung madalas ginagawa, grab sampling lang. So, it is suitable when analyzing situation at a specific site. So, kung kunyari gusto lang natin malaman, yun nga, kung trend lang, so usually grab sampling na yung ginagawa natin. Analyzing for unstable parameters that have to be measured right away. So, kunyari, dissolved oxygen, temperature, pH, salinity. So, yan kasi hindi na pinapa-analyze yan sa lab. Usually, kung meron kayong mga equipment, ito-test na siya on-site. So, mas maganda kung grab sampling yung gagawin. A snapshot of water quality of a particular instant is desired. The characteristics of water are known to be relatively constant over time. Collecting samples to be analyzed for parameters that could be adversely affected by compositing process. Okay. So kung may gusto lang tayong malaman in particular or time ng mga kunyari kung nag-discharge yung isang company nga doon sa lugar, we can conduct grab sampling dun sa water body. So usually grab sampling is enough kung hindi naman gano'n nagbabago yung quality ng water body or kung same naman yung mga dini-discharge dun sa lugar na yun. Now, composite samples naman, preferred yan kung assessing the total concentration of substance or pollutant in water or population of or the population of organism. So, kung medyo paiba-iba yung dun sa concentration ng pollutants dun sa water na yun, pwede tayo mag-conduct ng composite sampling. The variables to be assessed are unevenly distributed. Yun nga, kung medyo iba sa umaga, iba sa gabi, yung dini-discharge o yung quality ng water body nagbabago, pwede tayo mag-conduct ng composite sampling. Okay, so gaya na banggit ko, there are different types of composite sampling. First, yung fixed volume composite sample. So yun yung fixed yung volume nung kinukuhanan natin. Both the time interval and the size of the sample remain constant. So this is used when flow rate of the water does not vary more than 15% of the average flow. So fixed volume, so ito yung normal na may time interval lang, kunyari per hour, kumuha ka na sample. Tapos, same volume ng sample yung kinukuhanan mo. Tapos pinagsasama-sama kasi siya. So kunyari, kukuha ka ng 100 ml ng volume ng water ng discharge sa isang lugar, tapos within 24 hours. So tapos every hour kumukuha ka ng sample within 24 hours. So yun yung fixed volume composite sample. Time composite sample, this is collected by mixing samples of equal volume collected at regular time interval. So, okay, so ito naman, equal volume naman, different time regular time interval din. So, time composite sample. So, kukuha ka na sample, dun nga, equal volume sa regular time. So flow proportion composite sample. The volume sample collected is a proportion of the flow rate. So ito nakadepende siya sa flow volume mo. So kung paiba-iba yung flow nung dini-discharge, so iba-iba naka-ratio doon, naka-compute doon kung gaano kadaming volume kukunin mo. Depth integrated composite sample, this is collected predetermined depths of the water column, equal sample volumes and mix in water to one container. So, ito usually kinukuha nito sa ginagawa ito sa mga water bodies. Kung gusto natin malaman kung yung holistic water quality from yung surface niya, dun sa middle area niya, tsaka sa bottom area ng water body. So, pwede natin kukunin per depth yung sample tapos pagsasamahin. Okay, so prior sa pag-conduct ng sample, note lang, please make sure that you wear the proper PPEs. So especially kung pupunta ka sa isang facility, like industry facility, syempre, kailangan, alam mo din na hindi basta-basta pumapasok doon. And for your safety, kailangan either meron kang hard hat or mga wader. Pero kung water river naman, or water bodies yung kukolektahan, usually gloves, face mask, tsaka probably boots kung medyo malalim yung lugar, tsaka life vest kung magbabangka ka, or wader kung nasa coastal ka, kung kuha ka ng sample, especially kung doon ka na sa mga nasa chest level na area, or mas malalim pa, so mas maganda meron ka mga ganitong personal protective equipments. So magandang nakikipag-coordinate, especially kung papasok kayo sa isang facility, kasi nga usually sila na nag-prepare kung anong mga PPEs yung gagamitin na. So, isang important na factor dito, kapag magko-conduct ka na sample, make sure na dala mo yung proper appropriate containers depende sa parameters na kukolektahan mo. Kasi minsan nakaka-effect yan. Minsan hindi pala pwede sa plastic kung magko-conduct ka ng sampling na ipapatest mo. For example, nutrients, hindi pwedeng plastic yung water container mo. So, importante na magdadala ka ng right container at saka yung right volume para make sure na enough yung water yung sample mo. So, dito dun sa manual namin, nandoon kung gaano kadami yung requirement, kung anong mga containers, at makikita mo na dyan kung anong mga preservation requirement at saka holding time. So, dun sa table na yun, makikita mo, kunyari, for example, if you're going to conduct tests for ammonia or nitrogen, kailangan, pwede siyang glass, pwede siyang plastic, pero usually kailangan wide mouth yung kukuhanan mo ng sample. And then, kailangan at least 400 milliliters. Kapag oil and grease naman, ito kailangan glass lang. So kung kailangan glass yung container mo, tapos wide mouth, tapos kailangan at least 1 liter ng sample yung gagamitin. Nandito din pala yung mga analytic method, pero usually sa mga laboratory na yan, sila na may concern kung anong mga methods for analysis nila. Then for VOD, kailangan din 1 liter at least. Tapos pwede siyang plastic or glass. So para ma-determine mo kung anong mga containers ang pwede sa kanila. Pero usually meron diyan ng mga parameters na pwede naman pagsamahan. So for example, yung BOD, usually kino-collect siya with TSS kasi halos pareho lang naman sila ng container requirement. So more than, kapag ganun, pinagsasama-sama yung isang parameter, ay yung samples for different parameters na itetest, usually mas madami kaysa sa volume requirement nila yung kinukuha natin per container. Like for nitrates, ammonia, phosphates, yung mga nutrients, isang container yan. Tapos BOD, TSS, and color, isang container. And then yung coliform, yan yung mag-isa. Fecal coliform. So 100 ml lang. Kailangan sterilize. Tapos glass. And then, tapos kailangan mindful din tayo sa holding time. Kasi, syempre, kino-compute din natin yung travel time natin from collection of sample papunta sa laboratory. So kung kailangan, for example, yung BOD at saka ito coliform, kunyari BOD kailangan 6 hours na sa laboratory na. So mag-alat tayo, at least yung travel time natin dapat maximum na 4 hours. Kasi possible na may iba na yung result kung mapasok yung mga samples natin. And sa preservation, especially kung more than 6 hours yung travel time natin, kailangan talaga, minsan ina-acidify siya. Like for example, sa ammonia and nitrogen, kailangan mayroon na siyang, ilalagyan mo na siya ng sulfuric acid kapag itatago na yung sample for preservation kung ita-travel na siya. So, sampling methods, meron tayong yung ginagawang pag-collect na sample na direct method at saka yung syempre yung sampling with intermediate container. So, direct method, yun nga, direkta from the source tayo kukuha. Kung kunyari sa effluent, usually nga dun sa outfall or dun sa kung kukuha tayo ng sample sa septic tank, dun tayo kukuha. Pero hindi kapag effluent, hindi ko ina-advise yung direct method kasi nga hindi rin natin naman alam kung ano yung laman nung ini-discharge ng wastewater. So preferable yung using intermediate container. Usually kapag sa mga effluent or sa mga wastewater tayo, kukuha ng sample, usually bucket method, yung kukuha tayo ng sample sa isang bucket. At doon tayo kukuha, doon natin ito transfer sa ating mga sampling containers. Kapag naman water body, dito naman madalas direct method na rin or bucket method din. But yun nga, kung kukuha tayo sa mas malalim na area ng water body, merong mga equipment dyan like yung van der Waals para mas accurate yung depth ng kukuhanan natin ng sample. Okay, kung direct method yan, sa mga sampling, kung kukuha na tayo ng sample, okay, so ang pagkuha ng sample dyan, keep the container into the river face down. So yung bunganga nung container kailangan nasa ibaba siya. Siya yung unang didikit dun sa water natin, and then slowly lift the beaker towards the flow. So tapos yun nga, pa-diagonal yung shape. I-tilt natin siya pa akyat. So pataas. Fill the beaker up to 4/5 flow, and then water sample ready for analysis. So pero tayo kasi, ang ginagawa natin, ang determining natin dyan, kung sa container natin, hanggang neck. So hindi natin usual, especially kapag nutrients, hindi natin siya finifill up hanggang dun sa pinakabunganga. So hanggang dun sa may before nung rim nung river ay nung container, dun natin finifill up. Except kung kukuha tayo ng sample for BOD, mamaya makikita nyo. For BOD naman, and kung kukuha ka sample for DO, kung hindi on-site yung DO mo, baligtad naman. So i-dip mo na yung container, tapos bubuksan mo siya para pumasok yung water sample doon sa container. Okay. Ito yung kapag gagamit ka ng Van Dorn sampler. So usually naka-vertical siya, tapos pag ibababa na siya, mag-horizontal siya. Tapos hihilain mo siya. Tapos kapag nandun ka na sa depth, desired depth mo, hihilain mo siya para mag-close yung yung mouth niya para ma-collect yung sample. Okay. So kapag BOD, TSS, and heavy metals, usually plastic or glass bottle, pwede na. More than 1 liters nga, nabanggit ko, ang kinukuha na namin sample for analysis, kasi usually BOD and TSS, madalas magkasama yan sa isang container. Pero kapag magko-conduct ka ng BOD sample, make sure na airtight yung container mo at walang bubbles, walang air sa loob. So usually kapag makita mo mabula siya, pinipitik pa siya at tapos pinupuno siya hanggang dun sa cap. So kailangan punong-puno siya at wala talagang room for air dun sa container. Kung ipapa-analyze mo yung BOD niya. And then sealed it with the masking tape kapag nasara mo na siya. For nutrients naman, oil and grease, yun nga, usually wide mouth yung mga containers natin. And then yun nga, hindi natin siya finifill up hanggang dun sa bunganga, hanggang dun sa neck lang. Tapos, tinatakpan siya ng aluminum foil para hindi dumikit dun sa cap ng container yung sample. Tapos, sinisealed siya with masking tape. Now, for coliform, usually ang coliform, ang container niya naka-sealed na yan kasi nga sterilized yung container niya. Tapos, kapag kukolek ka ng sample sa container, Like so ilulublob mo yung container, dun mo siya bubuksan para hindi siya makontaminate ng mga bacteria sa taas. Pero near pa rin sa surface kasi kukuha tayo sample dun sa surface ng water. Tapos, ito lang medyo tricky pero sana yan lang din. Make sure na ganun din, hindi siya punong-puno kasi naglalagay pa sila ng mga preservation at saka mga reagents dyan kapag i-analyze. So, make sure hindi hanggang neck lang yung kukuha mo yung sample. Tapos isasara mo din siya dun sa, before mo siya i-lift pataas nung container nung water. Tapos, isi-seal. Actually, wala na siya dapat sa, hindi lang nawala yung bucket. Pero wala na siya dapat sa bucket kapag isi-seal mo na siya with aluminum foil and then masking tape. Tapos ibalik mo na siya sa dun sa resealable bag. It's important din na naka-label yung ating mga containers. Properly labeled sila. So, nandyan yung name of water body or yung institution kung saan siya kinuhanan, location natin, and yung saan yung lugar na pinagkuhanan natin, date and time of collections. For yung name, date, at saka yung time. Especially yung time kasi nga meron tayong holding time. Depende sa parameters. So, para maging guide rin ng analyst kung ipaprioritize nila agad na i-conduct yung sample doon. Name of person na nag-collect ng sample, syempre. Kung sino yung more responsible person na nag-conduct ng sample. Ngayon, you can choose to lagay din yung water quality to be analyzed. Kunyari for BOD, TSS, and color, kunyari yung papatest mo. And at saka kung ano yung mga on-site na test na ginawa. Kung kinuha mo na siya ng pH, dissolved oxygen, at saka temperature, lagay mo na rin. But, sa amin kasi practice namin, yung water quality to be analyzed at saka yung on-site mga parameters obtained, hindi namin na linalagay sa container. Linalagay na lang namin sa field data. Kasi, usually nababasa yung container. So, minsan mahirap na siyang sulatan kapag nabasa siya. So, minsan hindi na talaga siya nasusulatan ganun. So, basta maganda, available yung information na yun. So, enough na minsan name, source of location, date, time, at saka yung persons responsible for collecting na nandun. Para alam natin kung hindi naliligaw o nawawala yung mga containers natin. Okay, so water control natin, we need to make sure na yung quality nung samples natin hindi na co-compromise. So, we need to make sure na yung equipment are calibrated and maintained. And then we need to, kung kailangan natin ng quality control checks, usually hindi naman lagi, pero we need also mga field blanks na linalagay just to make sure na walang human error na nagagawa dun sa pagkuha natin ng sample, and dapat nga kung kailangan well-preserved yung mga samples natin. And then yung field data, yung nabanggit ko, finifill upan yan regarding dun sa condition ng sample natin, and chain of custody, pag-transfer sa laboratory. So this is example of yung field data form. So GPS ng lugar, time, kung makuha mo yung air temperature, cloud cover, weather condition, visual color of ng water, at saka other observations. For example, kung kunyari dun sa pinagkukuhan natin, kunyari dito hog farm. Tapos dun sa prior dun sa sampling site, dun sa discharge point nila, merong kino-construct, may construction na ongoing. Na possible na mag-contaminate dun sa kinukuha na sample. So pwede natin ilagay dyan. So kung medyo kakaiba yung sample results natin, meron tayong possible na explanation. And then nga kung nag-conduct tayo ng mga on-site analysis like dissolved oxygen, temperature, pH, ilagay rin natin. So minsan, dun sa other description, pwede rin natin ligay yung mga observation natin. Especially, siyempre hindi naman natin tinitikman yung mga sample, delikado yun. but minsan naamoy natin. So, dito pwede nating guide yung odor descriptor wheel ni McLeany and McLeany or 2002. Kung kunyari nakakaamoy tayo ng parang apple, yung amoy, cherry, so pwede natin lagay fruity yung odor niya. Kung amoy patay na isda naman, pwedeng lagay natin fishy yung odor. Kung amoy dugo or amoy septic tank, amoy suka or bulok na itlog, pwedeng lagay natin offensive yung odor niya, yung description natin. Para lang uniform yung description natin ng odor nung na-observe natin. Okay, so medyo naligaw to. Ito yung sa mga field blanks. So isa to para pag-check ng quality, pag-check nung quality nung sinasample natin just to make sure. So hindi naman lahat to pwede ginagamit lahat sa isang samplingan. Pero kung gusto lang natin malaman or ensure yung na tama yung procedure natin, we can use field blanks. Like equipment blank, okay, laboratory water fee of analyte is transported to the site and processed through the same sample collection device. Preserve is necessary and return to the laboratory for analysis. So, yung water ay manggagaling sa laboratory, dadalhin siya sa site, tapos same yung method ng pag-collect natin ng sample from yung site. Same yung mga sample devices natin. Kung itetest natin siya sa DO, ganun din ang gagawin natin sa water na yon. Tapos ita-transfer back natin siya sa laboratory. Ito nga, equipment blank. So, para make sure na walang error dun sa mga equipments na ginagamit natin. So, kung questionable yung mga equipments natin or hindi tayo sure dun sa mga ginagamit natin, kung hindi tayo sure kung calibrated, maganda, meron tayong equipment blank. Field blank is usually laboratory deionized water that is transported to the sampling site, open to the contaminated environment, and processed as sample. Filtration and preservation. So, field blanks, equipment blanks are type of field blanks din naman. But field blanks, so ito yung mga water din, yun nga, tinatransport sa site. Binubuksan siya para ma-experience yung mismong environment dun sa site na yun. Especially kung merong mga construction dun sa site. So, tapos i-analyze din siya. So, parang check and balance lang dun sa kinoconduct nating sample para malaman natin kung may effect ba yung mismong condition ng lugar dun sa sample na kinukuha natin. Filter blank or cartridge blank, laboratory water is used to rinse the filter and filtration apparatus. So, actually, hindi tayo gumagamit ng filtration sa pag-collect natin, usually na sample natin. Pero pwede to kung kunyari ang question natin yung kapag lininis na natin yung mga containers natin after sampling, pwede natin gamitin yung filter blank or cartridge blank para determine kung hindi contaminated yung pinanlilinis natin. Trip blank, ito kung may involved to sa mga volatile organic compounds na tinetest. So, usually, laboratory supply samplers with volatile organic compounds vial containing acidified analyte-free water. The vial is transported to the sampling site and returned to the laboratory without being opened. So, yung mga vials, dinadala lang siya sa sampling site, tapos sineturn din sa laboratory to check nga regarding to sa volatile organic compounds. Field duplicates. Ito are two separate samples collected at the same time and placed under identical circumstances and treated exactly the same throughout the field and laboratory procedure. So, usually field duplicates yung madalas na pang check and balance natin kasi nga just to make sure na hindi malayo yung mga analysis na ginagawa. So, Ibig sabihin, dalawang samples kinokollect natin per area, the same time, tapos same yung treatment, tapos same yung analysis din. Then split samples. Ito naman, kung medyo questionable yung nag-a-analyze, pwede tayong mag-conduct ng split sample. So, our aliquots of samples taken from the same sample container and through thoroughly mixed and compositing sample, they are analyzed independently and are used to document and interlaboratory precision. So, basically, dalawang sampling na gagawin na na ng separate laboratory analysis, laboratory institution yung magpo-conduct ng test. And then blind sample, ito actually ito yung pinakamadali. It's a sample submitted to the lab for analysis. The composition or origin of the sample known to be submitted or is unknown to the analyst. So ito, magdadagdag ka lang ng isang sample pa, dun sa isa dun sa mga random na kinukuha ng sample, papa-analyze mo siya, pero hindi alam ng laboratory kung anong origin niya. So, it's a blind sample. So, para i-check mo nun kapag dumating results kung same siya nung pinatest mo, nung actual na pinatest mo. So, ito mga quality control, field quality control checks lang. So, yun nga, you can choose kung anong pinakamainam to. Depende rin sa objectives nung sasamplingan. Okay, so, ito yung, kunyari, kung ka ng field blanks, so, the method, pre-clean equipment blanks, so yung mga equipment blanks, if contamination is suspected, 1 per sampling trip if equipment is not cleaned in the field, and 1 quarter per project. So, kung suspected mo nga may contamination sa mga equipments mo, pwede kang mag-conduct nito, yun nga, minsan sa isang sampling or per quarter. Depende nga sa dalas ng pag-conduct mo na sample. Pag field blanks, kung field blanks yung gagawin mo, hindi mo na kailangan ng equipment blank, or pwede ka rin gumawa nito 1 every 20 samples. So field duplicates varies depende sa project, or 1 every 20 samples. So hindi lahat ng sampling kailangan ng ganito. Yung nga, pag check lang naman ng quality siya, quality check. So para lang masigurado mo na meron ka lang support na tama yung procedures, tama yung laboratory analysis, at saka walang error din sa mga equipment sa ginagawa mo. Tapos sampling preservation and holding time. Kanina napakita ko na stable, depende sa parameter kung anong mga preservations yung gagawin. Pero kung within 6 hours, sa amin kasi, usually, kung agad namang nata-transfer sa laboratory yung mga samples, usually ina-ice yung mga ice box, and mini-maintain at least 6 degrees Celsius pababa. On the start of the time of the travel, hindi mo naman ma-maintain 100% yun. Pero on the start of the travel ng sample, kailangan 6 degrees Celsius. At least 60 degrees Celsius yung temperature dun sa loob ng chest. And then ito, yung nakakalimutan naman na nagkakonduct ng sample. Once na nakakonduct ka ng sample nga, rinse the outside surface of the sample with tap water. So before placing them into a cooler or ice chest. So kailangan malinis to avoid contamination din sa mga ibang samples natin. And then photo documentation, parang kasi nga pwedeng gamitin to as evidence or in support sa study mo na tama yung ginagawa mo. So take photographs when there are naturally occurring landmarks such as large trees or boulders para madaling ma-identify. And also, kuha ka ng sample mula sa pag-collect mo ng sample hanggang sa pag-transfer mo sa mga sampling containers, hanggang sa pag-test mo ng on-site, hanggang sa pati dun sa paglagay lang ng masking tape, pag-transfer dun sa chest, maganda naka-document yan. And maganda geotag yung mga photos mo para malaman din kung saan siya kinuha at kung anong oras. Then yung chain of custody, ito na yung pag-fill up ng form para ita-transfer mo na siya sa mag-a-analyze dun sa sample. So ito yung example namin ng chain of custody na ginagamit namin. So nandyan yung client or source, tapos yung address, kung sino nag-sample, tapos sino nag-submit, tapos kung yun nga kung anong source, tapos kung anong mga station mo, identification station, kung anong type ng sample, kung kailan at saka anong araw siya sinamplingan, kung anong araw at saka anong oras siya sinubmit sa laboratory, and kung anong mga pinapa-analyze mong parameters. So, it's important na meron chain of custody para nga pati yung mga oras naka-document and kung anong oras siya tinanggap ng laboratory. Siyempre, kapag yan, kasi minsan yun nga, kapag napapaso yung sample, baka kasalanan mo ba or baka yung laboratory hindi agad inanalyze. So may ganda meron kayong chain of custody. Nakalagay din pala dyan kung anong mga field preservation yung gagamitin. So in summary, you need to plan ahead, follow your plan, strictly follow the sampling procedure, and properly document what you did. So, and kung kailangan nyo ng references regarding dito sa diniscuss ko, yun nga, madali ito makita online, yung mga water quality monitoring manuals namin for ambient and effluent, yung volume 1 and volume 2, at saka yung DNR Administrative Order number 8 series of 2016, yung water quality guidelines and general effluent standards. So, nandyan yung mga link, madadownload nyo siya yan. So, yun. So, yun lang for water sampling. Thank you very much. May questions ba?
Thank you for sharing your expertise with us today, Sir Ramjay. For the questions po, pwede po nating isend sa chat box ng ating Zoom, or para po sa ating mga FB live stream viewers, pwede po tayo mag-comment sa ating FB live stream video. For the continuation of our session, may I call on Mr. Al D. Makibo. Sir Al. Sir Al, we can't hear you po. Wala po, sir. Wala po. Habang nag-aayos po si Sir Al, pwede po nating i-share yung ating Facebook live stream para po makapanood din yung iba. Wala pa po, sir. Wala pa po. Wala pa po, sir. Habang nag-aayos po si Sir Al, sa mga questions po, pwede po nating isend sa Zoom. State your, ano na lang po, yung institution, kung saan po kayo affiliated. Tsaka habang hindi pa po tayo nagpo-proceed sa ating second lecture, sinend po ng isa sa ating technical support team yung attendance form. Para po sa mga hindi pa po nakapag-attendance, pwede pong mag-attendance. Gising pa po ba yung mga participants natin? Kung gising pa po, pwede po tayong mag-thumbs up. Show us your thumbs up. Yan. Meron pa. Yung mga nagta-thumbs up. Well, naka-unmute po yata kayo, sir. I'm sorry, naka-mute po yata kayo. Naka-mute po kayo, sir. Naka-mute po kayo.
Kanina kasi pwede eh.
Wait na sir, ayaw?
Hello, sir? Wala pa?
Meron na po. Yan na po.
Ay, meron na, sir.
Yes po.
Okay, sige. Okay. Thank you, sir.
Okay po.
Start na ba, sir?
Yes, sir. Pwede na po kayo mag-start.
Okay, good morning po. So I will just share my screen. So good morning po sa ating lahat. First of all, I would like to congratulate yung mga PS Bio third year for reaching this far. So konti na lang mag-graduate na kayo. So ito. So ito yung ginagawa namin sa BFAR, sa Bureau of Fisheries, specifically doon sa project na Balik Sigla sa Ilog at Lawa. So yung water quality assessment of inland bodies of water. So water, ano ba yung water? So water is basically the environment of fish. So hence it is whatever its characteristics are and whatever happens to it affects the fish greatly. So the Philippine inland waters, so meron tayong 211 lakes. 79 dito ay natural or major lakes. Then meron naman tayong 421 rivers. From it, meron 18 major rivers, kasama dyan yung Pampanga River. Then meron tayong 22 marshes and swamps and more than 500 square meter of water reservoir. So the Philippine wetlands, meron tayong 1,616 species of aquatic flora and more than 3,600 species naman ng fauna. So, noong 2013 yung data na yan. So, pwede nagbago na yung data na yan as time goes kasi meron na yung mga bagong discovery ng mga inland bodies or mga species din. So, pag-aaralan natin yung background ng lake and river. Dito kasi sa Bureau of Fisheries, especially yung project na hawak ko dito sa BFAR, is concentrate siya sa inland bodies na katulad ng lake or lawa, rivers, and yung mga reservoir. So ano ba yung lake? So bigyan natin ng konting review. What is lake? So lake is a large inland body of fresh water surrounded by a land or mountains. So malaki siya, malalaking inland bodies of water yan. So meron siya, nagkakaroon ng lakes dahil sa paggalaw ng lupa, pagputok ng bulkan, at yung sa mga malalamig na lugar naman, doon yung pag natutunaw yung tubig doon sa mga malalamig na lugar, yung may mga yelo o yung mga glaciers. And sa tao din, meron kasi mga man-made lake. So most lake on earth are fresh water. So matabang ang tubig mostly sa mga lakes natin. Pero meron din naman may mga alat. or merong salt water. So lake is a lentic water. Ibig sabihin, hindi gumagalaw yung tubig, no? Sa lentic. So ano ba yung sources ng lakes natin? So yung sinabi natin kanina, glacier activity. So yung mga yelo sa sa taas ng ating earth is na ano siya pag natutunaw, then yung mga depressions doon ay mapupuno sila ng tubig, so nagiging lake yung mga yon. So tectonic activity, nagkakaroon ng paggalaw sa lupa. So nagkakaroon ng mga depressions or mga basins. Tapos ayun, pag nalagyan ng tubig, nagiging lava din. Then volcanic activity. So after erupting, so some volcanoes collapse, forming basin that collect water. So parang yung Taal yata, ganyan yung nangyari sa Taal volcano or Taal Lake. So lake coloration. So ito yung mga ano parang mga ano natin sa lake coloration. So it can be varied from blue to brownish red. So color largely depends on the chemistry and the biology of the particular lake. So sinasabi natin kapag daw shallow o mababaw yung lawa or yung lake, tapos maraming phytoplankton, and tapos may large quantities of nutrients, deep green yung kulay. So yung nasa left side ng ating yung picture. Then kapag naman daw malalim yung lake, tapos konti yung quantities o yung dami ng nutrients, tapos mababa din yung plankton count, clear blue naman yung kulay ng ating lake. So, ito ay lahat ay review muna dun sa gagawin natin mamaya. So, the lake properties naman, so meron tayong chemical properties. So, ano ba yung mga kinukuha natin sa chemical properties? Usually yung the oxygen and nutrients distribution na affect din sa lake's health. Then the next one is the physical properties. Lakes are not uniform mass of water, so hindi pare-parehas yung dami ng tubig sa isang lake. So iba-iba yon, magkakaiba. Hindi rin pare-parehas yung lalim, hindi rin pare-parehas yung shape, at yung temperatura ng tubig ay hindi rin pare-pareha. Same din yung kanilang sediment, o yung sa ilalim ng or yung sediment ng isang lake is hindi rin pare-pareha sa bawat lake. So depende yun sa location. Then the biological properties naman is meron community of animals, plants, and microorganisms which interact and depend on each other and the environment in which they live. So yun yung sa lake properties. So ano ba yung bakit ba mahalaga yung lake, no, sa tao or sa ating kalikasan? So meron siyang social contribution to social economy kasi. So yung ibang lake kasi ginagamit din sa iba't ibang pang-araw-araw na gawain. Iba naman livelihood and fishing. Livelihood and fishing, pag-fish or paglagay ng mga fish cages or fish pens. Katulad ng Laguna Lake, Taal Lake, may mga fish pens dyan at fish cages. So tourist and recreation locations. So meron isang pinakasikat na lake na under ng Basil program is the Sumlang Lake po sa Region 5. So kung meron kayong time at Google niyo yon, ang photo bomber kasi nung lake na yon is the Mount Mayon. So ginagamit din kasi yung lake for tourist and recreation. Then biodiversity conservation areas. So meron mga endemic species na nabubuhay dun sa mga particular lakes like the Taal Lake na yung freshwater sardines or yung Sardinera tawilis na doon lang nakikita. So kino-conserve din yon ng authority doon sa na humahawak sa Taal Lake. Then the natural balance preserving reservoir. So ano naman yung river? So yung river naman is malaking large quantity of lotic freshwater. So itong sinasabing lotic is moving. So gumagalaw yung tubig sa river. So river begin at their sources in higher ground sa mga mountains or sa mga hills where rainwater or melting snow collects the form of and forms tiny streams. So the great majority of rivers eventually flow into large body of water like an ocean, sea, and large lake. So yung dulo kasi ng mga river usually ay dagat dahil dun pupunta sa pababa sa sea level, then yung iba naman sa lake level. So ano ba yung mga sources natin ng lake? Ay ng river. So spring, sa taas ng bundok yan. So begins a natural spring releases from the underground. So pwede rin ng rainfall or melting snow. Some river begin in the mountain or hills where rain or water or snow melts, collect, and form small channels. So the next is lake. Kasi yung ibang mga lake, meron silang runoff in the form of river. Kailangan kasi, minsan kasi hindi, yung dike kasi ng river, para ma-support yung hindi masira yung dike ng lake, is na kakaroon siya ng maliit na river para yung sobrang tubig is mag-flow. So ano pa yung contribution ng river sa social economy? So yung river, iba, ginagawang source of drinking water. So abundant supply of water is a prerequisite for a human settlement. So dito sa atin sa medyo urban areas, medyo hindi na to ginagawa kasi polluted na yung ating mga river. Then river and crop irrigation. So in agriculture, irrigation plays an important role. So usually sa mga agricultural areas like bandang in Nueva Ecija, Tarlac, yung mga yan is river ang pinagkukuhanan nila or mga patubig. Kumukuha sila doon ng pang-irrigation. Iba naman sa mga fish pond dito sa Pampanga. Then river as power source, so industries need power which is being generated at a large number of hydroelectricity. Water transport, yung river kasi ginagamit din siya na transport areas. So may mga bangka and ayun sa parang Pasig River. And dito sa Pampanga, iba nagbo-boat sa papunta ng ating mga coastal communities sa may Masantol, Macabebe, papunta ng Apalit, and some parts ng Bulacan. So river as source of livelihood. So yun, iba, nakapagtayo sila ng mga fish cages and iba pang fishing talaga. So ito yung status muna ng lake sa Philippines. Rivers and lake in the Philippines. So 36 major lakes, Philippine lakes in the Philippines. So 55.5% dito ay threatened. May 41.7% are in good condition and 2.7 are in critical condition according to Guerrero 1999. So malaki yung nawawala sa kita ng ating pamahalaan dahil sa mga to. So almost 67 billion, no? And 17 billion dito is sa fisheries production. Madami yung tao, kumukonti yung production ng isda. So, ayan. So by 2025, so 1.8 billion people will be living in countries or regions gripped by water scarcity. So 2/3 of the world's population could be facing a possible water shortage. Sabi ng United Nations. So only 1/3 of 36% of the Philippine river systems are considered suitable for public water supply. Dahil yan pa rin sa water pollution. So hindi ko na ito di-discuss, pinapakita ko lang sa inyo yung mga status ng rivers sa atin dito sa Philippines. So ano ba yung mga issues ng ating mga lakes and rivers? So the pollution, water pollution, so syempre may mga dumping of mga sewage, pesticide, and fertilizer residue. Meron naman tayong mga namatay na mga halaman at mga hayop. So yun yung nagkakos ng water pollution dumping of solid waste and not segregating. So usually problema ito sa mga rural areas and urban areas also, sa mga illegal settlers. Human activity, so nagkakaroon tayo ng problema sa water pollution dahil sa human activities, number one, dahil sa illegal logging, indiscriminate quarrying, land subsidence, and squatting and unaggregated land use. Then, staggering increase of population. So, nagkakaroon kasi ng establishment, maraming factories, recreational centers, kaya dumadami yung mga consumption ng tubig. So, ang ending, nagkakaroon ng water pollution. So, ito isa sa reason kung bakit nagkaroon tayo ng Baliksigla sa Ilog at Lawa program o Basil program ng Bureau of Fisheries and Aquatic Resources. I will be discussing the lake profiling, including the yung pagkuha ng water samples, kung paano natin siya tinitest, yung kanyang water quality. Pero hindi siya limited doon lang. Meron pa siyang ibang areas. So sa Basil kasi, we have 4. The lake profiling, social preparation, lake rehabilitation, at saka lake management. But I will be just discussing itong lake profiling. So ano ba yung ginagawa natin dito sa lake profiling? So lake or river resource profiling, so this involves the scientific study on the actual condition of the lake and river systems and other ecological consideration. So kinukuha natin yung lakes and river resource profile. Kasi kailangan natin siya for the baseline assessment, no? Yung pagkuha ng physicochemical parameters and plankton assessment. So dito na tayo pumapasok sa water quality assessment. So ano ba yung mga parametro or parameters na kinukuha natin sa tubig? So number 1 is the temperature, number 2 pH, 3 salinity, 4 dissolved oxygen, 5 turbidity, at number 6 is the ammonia. Actually, parang ano na to dun sa kay Sir kanina sa EMB na discuss na ito, pero bigyan na lang natin ng konting discussion lang kung paano siya kinukuha. So number 1 muna dun sa list natin is the temperature. Temperature is defined as the degree of hotness or coldness. So water temperature varies according to season. Siyempre nagkakaroon pag mas mainit, siyempre mas mainit ang tubig. Depth, lalim, then time of the day. Siyempre mas mainit kapag mga bandang alas 10, alas 11 hanggang alas 2 ng hapon, then pababa na ng mga bandang 4 hanggang gabi. So influences the kind of organism that can live in a river or lake. So siyempre kapag yung isang body of water is mas mainit, meron lang mga certain organism na pwede mabuhay doon dahil hindi lahat hindi lahat ng organism ay mabubuhay doon kapag mainit. So yung iba kasi ayaw din sa masyadong mainit. So ano din sila, very sensitive yung mga isda. So affects other physical and chemical parameters of the water. So sinasabi natin sa study, 25 to 32%, 35 to 32 degrees Celsius, yun yung optimum growth ng isda, no? Sa aquaculture, yan ang ano natin sa aquaculture. So ang ginagamit na pagkuha ng temperature is the thermometer. Pasok siya sa tubig. The next parameter is the pH or the power of hydrogen. So ano ba ito? So mini-measure niya it measures the acidity or basicity of the water. So most freshwater species, ang pH na requirement niya or ideal for growth is 6.5 to 9. So nandun siya sa in between 7. So, 7 is the neutral. So ito, nilagay natin dito yung levels kung saan dito pag less than 4, kasi pag 4 kasi is acidic na yon, so parang yung suka, no? So 4 ang pH ng suka, so acid depth point. So 4 to 5, no production. Ibig sabihin nun, hindi yung isda is hindi siya mag-reproduce pag 4 to 5 yung pH level. Then 6.5 to 9 to 9 pH is yan na, yung desirable range for fish production. Then 9 to 11, slow growth. So hindi mabagal ang paglaki ng mga isda sa aquaculture pag 9 to 11. Then pag 11 pataas, so alkaline at that point, masyadong mapakla naman yung tubig pag ganyan. So in fish ponds, sabi nila, the low pH levels can accelerate the release of metals from rocks and sediments which affect their metabolism. So ang pinangkukuha nating pag-gauge sa pH is the pH meter. Next is salinity, the amount of salt dissolved in a body of water. So as salinity increases, the solubility of water, of the oxygen in water is decreases. So sinasabi pag mas maalat, yung dissolved oxygen is mas bumababa. So ito. So dito sa atin, pinapakita natin na 0 to 0.5 ang considered as fresh water pa rin. So sa mga ponds, lake, rivers, and streams. Tapos sa brackish water, dito kasi sa brackish water or yung estuaries, yung sing sinasabi nila. So dito mostly yung marami ang nabubuhay at pwedeng i-culture. Ito yung mga magiging pag-culturean talaga yung sa brackish water. And mga high value species. Then ito nag-range siya sa 0.5 to 30 ppt or parts per thousand. So maraming diyan sa brackish water. Maraming pwedeng i-culture. So pag more than 30 or 30 to 50 saline naman na yun. So sa mga sa ocean or sa dagat na yung alat na yun. So ang ginagamit natin na pang-measure ng salinity sa tubig is refractometer. May sinabi si Sir kanina dun sa EMB na hindi tinitikman. Actually, yung mga farmers namin, meron silang na-device na procedure. Tinitikman yung tubig para kapag nagko-culture kasi ng pag nagko-culture ng Pineus monodon or yung black tiger prawn at saka po ng baname, Pineus baname naman, is tinitikman kasi kailangan ng may alat ng tubig. So pag tilapia naman, dapat walang tubig. Pero kaya naman kasi ng tilapia buhay naman ng may tubig, ay na may alat, pero dahil wala man silang refract kasi medyo mahal yata ito sa market. So tinitikman na lang nila. So at naka-quantify nila actually yung alat ng tubig. The next one is the dissolved oxygen. This one is the most important na kinukuhang parameter sa tubig. So, ano ba yung dissolved oxygen? So, oxygen available in the water is dissolved oxygen or in dissolved state. So, DO. So, low levels of oxygens are often linked to fish kill incidents. So, yung usually kapag nagkaan yung aming office, pagka nagre-report yung mga farmers or for a particular area sa mga inland bodies, usually ang nagiging suspect talaga is low level of oxygen. Mamaya bakit kaya siya nagkakaroon ng low level of oxygen? Kasi baka sobrang dami nung na-stock doon, maraming nakatira na organisms or isda. So kapag gabi kasi nagkakaroon sila ng competition sa pagkuha ng oxygen. Even yung mga halaman kasi hindi sila nag-undergo ng photosynthesis sa gabi. So nag-consume pa rin sila ng oxygen. So nag-aaway-away sila doon sa available na DO. Sa gabi kasi walang production usually ng oxygen dahil nga walang araw. Then in general, saturation level of at least 3 to 5 milligrams per liter is required. So ideally, dapat nandito sa range na to yung DO level. So dapat nasa 3 to 5 ppm. So ang ginagamit natin pong pang-measure niyan is the DO meter or dissolved oxygen meter. Next, ito yung pwede naman kung i-send yung presentation na to. So ito yung mga species tapos yung nila oxygen na demand nila no sa mga sa aquaculture actually ito. So ayan yung mga range ng dissolved oxygen. Next is the turbidity. Ano ba yung turbidity? Yung labo. So refers to the relative clarity of the water. So it affects other physical and chemical parameters of the water. So materials can causes water to be turbid, include clay, silt, algae, plankton, and other microscopic organisms. So greatly affects life penetration. So paano ba to? Yung turbidity, so kinukuha natin yan. Ito kasi sa turbidity, iba-iba nga yun. As I said, na may algae, may plankton. So ito kasi yung mga natural na pagkain ng mga isda doon sa isang ecosystem, sa isang aquatic ecosystem, ano. So ang ginagamit natin pagsukat sa turbidity, sa fish pond at saka sa mga inland bodies is itong Secedis transparency. Paano ba yan ginagamit? So yung Secedis, itong kung nakikita niyo yung pink na yan, is tali yan. Then yung dis, mabigat kasi yan, lulubog mo yan. So yung iba, ginawa ko noon, kapag ako ay nagkukuha ng transparency, lalagyan mo na lang siya, meron siya dapat medida or panukat doon. So, ibababa, dahan-dahan ibababa, at dapat ginagawa pala ito during daytime. So start ka ng mga alas 8 hanggang alas 12. So para hindi ka bias. So ibababa dahan-dahan yung Secedis. Then pagkababa ng Secedis, hihintayin mong mawala yung hindi mo na makita yung plate. Hindi mo na makita yung plate. So kukunin mo ngayon yung reading. Kunyari nawala siya sa 35 or 40 na lang, nawala siya dun sa 40 na measurement. So i-mark mo yon sa 40. Then dahan-dahan mo ulit siyang iaangat. Then kapag lumabas na ulit, nakikita mo na ulit yung Secedis, kukunin mo ulit yung reading. So kunyari kung 40 yung kanina, tapos at 35, ano na siya, kung nakita mo na siya, pag-add mo ngayon dalawa. So 40 plus 35 is 75. Divide 2, that is 37.5. So ang normal kasing range natin sa Secedis ay 35 to 40. Yun yung nandun yung maganda, ibig sabihin maganda yung natural productivity or maganda yung meron mga natural na pagkain yung mga isda doon. So kapag masyadong malabo or masyadong yung mababa yung reading, ibig sabihin ng masyadong turbid, masyadong malabo yung tubig, maraming plankton or baka disturb din kasi yung place na nagalaw. So ayan, pag naman umabot ka na ng 50, 100 ganyan, hindi mo pa rin nakikita hindi pa rin nagdi-disappear yung Secedis, masyado naman malinaw nun. Ibig sabihin naman nun, walang gaanong natural food, no, walang kakainin yung mga isda doon sa tubig. So yun anong ginagawa sa fish pond kasi mina-manage talaga. Pero sa amin sa inland bodies of water kasi we'll just collect data na mababa nga yung rating ng turbidity. So yan yung Secedis. Yan yung Secedis transparency. So ammonia. Ammonia is actually the second most important water quality parameters after ng dissolved oxygen. Ano ba yung ammonia? So ammonia is a colorless gas with a distinct odor. Yung amoy na sinasabi na mabaho ang amoy ng ammonia kasi ito ay composed of nitrogen and hydrogen atoms. Causes nito is toxic effects on aquatic life. So highly toxic ito. So actually there are two forms, yung unionized ammonia, NH3, and the ammonium ion or yung NH4. Ang NH3 is the highly toxic, pero yung NH4 is harmless naman. So ano ba ang cause ng ammonia? Ang cause ng ammonia kasi sa fish pond, usually kasi kapag nagpapakain, yung mga uneaten feeds na meron, uneaten feeds is nabubulok doon sa ilalim. Tapos, syempre, magkakaroon ng bacterial, yung bacteria is kakainin, bubulukin niya yon, and some other factors. Magiging parang, imagine, dun kumakain, dun tumatae, umiihi, lumalanghoy, nabubuhay yung mga isda. So nandun lahat doon sa area na yon. And sa lake, for example, nandun din lahat. So ang ammonia is, and also yung mga plankton, eventually kasi magda-die off yung mga yan. Pag hindi sila na-consume, magda-die off sila. And it will cause also ng pwedeng mag-add doon sa production ng ammonia. Next. Ayun. So ang ang pinang-test natin ng ammonia is, iba-iba kasi yan. So yung ginagamit namin dito is bigay siya ng FAO or Food Agriculture Organization ng United Nations, ammonia test kit, yung hack kit. So isa colorimetry yung ano niya, yung procedure niya. So dun sa kung nakikita niyo yung dalawang test tubes na yan, no? So yung i-fill mo siya ng tag 5 ml, i-fill mo siya ng tag 5 ml ng tubig kung saan yung samples mo, i-fill mo siya, then maglalagay ka ng itong packet na to, para siyang reagent, so ilalagay mo dun sa isa, then mix well, for 3 minutes, stand mo siya ng 3 minutes. Then, after 3 minutes, lalagay mo yung isang packet ng reagent dun ulit sa same test tube. Sa same test tube. Then, for 15 minutes, stand mo siya for 15 minutes kasi dun mo abangan yung magiging reaction niya. So, paano siya i-read? Ito pong disk na ito, itong color disk na ito, ipapasok mo dito sa loob, then i-compare mo siya. Kung nakikita niyo yung dark green na yon, ibig sabihin ng mataas yung ammonia noon. Ang ating kasing, yung range lang ng ammonia is less than 0.2 sa reading. So meron niya reading doon. So dapat nandito ka lang sa may yellow. Doon lang ang reading mo. So mag-react naman yan. So depende din sa sample. So para hindi na kayo mahirapan, ang ginagawa, meron naman multi-parameter test, o yung multi-parameter water quality meter na available na sa market. So meron din kami niyan dito sa Bureau of Fisheries. So ginagamit namin ito, yung diniscuss natin kanina, thermometers, like this, refractometer. So ito, all-in-one na siya. Na pag inilagay mo sa tubig, is i-read na lahat halos yung mga parameters na kailangan mo. Except for the ammonia, wala pang ammonia ito. Pero parang merong modelo na susunod dito na meron ng ammonia. So ito, ilalagay itong probe niya, no? I-soak lang sa water, and then yon, makikita mo na yung reading dito. So pwede itong i-save, then balikan mo na ng after ng field work, or pwede mo rin naman kunin na lang siya as it is yung reading. So para mas madali yung buhay. So paano naman ang ginagawa namin sa plankton assessment? Phytoplankton, zooplankton, then yung mga isda kakainin yan, then so on. Paano? So phytoplankton muna tayo. So phytoplankton assessment. So primary source of energy in most aquatic ecosystem. Sila kasi yung autotrophs sa tubig. So uniquely adapted to the specific depths, habitat, nutrients, and chemical conditions in which they reside. So they are also the indicator of biological condition of the site. Very important ang phytoplankton because ito yung kinakain ng mga ibang malalaking isda or malalaking invertebrate sa loob ng aquatic ecosystem. Also, yung zooplankton, sila yung unang nagko-consume ng phytoplankton. Sila yung primary energy source. Next is the zooplankton. So zooplankton is a small, free-floating aquatic microorganisms. They are the microanimals. It can be yung isang stage ng invertebrates. Pwede siyang maging zooplankton, like yung mga crustaceans. So transfer of energy from phytoplankton kasi sila yung sila yung mostly nagko-consume ng phytoplankton to a larger invertebrate predators and fish. So pag kinain ng zooplankton yung phytoplankton, so sila yung first level ng trophic level, and then kakainin sila ng bigger fish. So para magkakaroon ng balance sa food chain ng aquatic environment. So it is also an indicator in change in nutrient pollution over time. So paano ba yung ginagawa namin sa pagkuha ng plankton? So una, yung sa study area, syempre, check namin doon. So pwede yan mag-Google sa Google Earth nga makikita niya yung area. So maghahanap ka na ng sampling sites. So usually 3 or 2 pwede na. Pero kami sa Bureau of Fisheries kasi 2 lang yung pinagkukuhanan namin. So depende sa laki ng area. Kasi kung pwede isa na lang ang pagkukuhanan namin. And 2 trials per station. Or pwede rin yung composite sampling na ginagawa namin. And also, pagdating kasi minsan dun sa pagpunta kasi namin, kahit kunya example ganyan siya kalaki, minsan kasi especially during the summer months or the dry season natin, nagkakaroon ng lumiliit yung water area. So nag-a-adjust kami doon ng sampling site. So paano yung methodology ng water samples? So sa phytoplankton, sa plankton assessment muna. So gumagamit tayo ng plankton net. So yung plankton net ay special net siya na may siya ng mesh size ng screen. So this one is parang medium size ito ng plankton net. And then ilulubog siya sa tubig. Dapat may clearance sa baba. Ito mabigat ito sa lulubog. Kailangan meron ito. Ito kasi yung sasapo ng tubig na magpapass through dito. So dito mo kukunin yung sample. Ito sa may tip na to, doon sa puti magkakaroon yan ng tubig. Diyan mo kukunin. So parang may gripo dyan. Then doon mo i-collect yung makukuha dito. So lalabas lahat dito. May iwan yung mga plankton sa loob. So kukuha ka sa ilulubog yung tubig. Dapat may clearance sa ilalim. Hindi ito dapat sa tip sa sediment o sa ilalim natin sa bottom. Hindi siya dapat mag-kiss or magdidikit doon sa ilalim. So ayun. Kuha ka ng desired amount. Usually meron naman kami sampling bottle na pinagkukunan at pupunuin lang. Then the physicochemical parameters, hindi na namin ito dala-dala sa site kasi napaka-hassle na magdadala ka ng ang dami nito, parang 5 na equipment. So ang dinadala na lang namin is the multi-parameter test kit. So ayun. So paano namin ginagawa sa plankton net? So ilo-lower daw siya sa 1 meter below the water surface kung malalim yung tubig. Then at the sample bottle namin, so yung pagkakuha, so water will be dispensed to 50 ml pre-labeled bottle. Immediate ang pag-analyze immediate po ang pag-analyze ng plankton. Pag medyo malayo yung area at hindi agad makaka-uwi within 24 hours yung sampler, nilalagyan na lang namin ng iba ginagawa ng Google solution, pero dito sa amin nilalagyan ng 10% buffered formalin. So ginagawa yon para ma-preserve yung mga plankton doon. Pero para mga 1 month lang siyang pwede. Kasi eventually kahit na may formalin, nasisira pa din yung structure ng plankton at hindi na daw mabasa ng attendees. So ayun. Also pati pala yung sa water quality, meron kami kasi insight yung drug sampling na sinasabi ni Sir kanina, meron ginagawa namin yan. Also nagkakaroon din kami ng duplicate test for the laboratory. So ilalagay din namin sila dito sa water sample, sa bottle na to. And then siya ng yelo sa isang cooler, lalagyan namin siya ng yelo papunta ng lab. So yun, then ipoprocess na siya dito sa laboratory na. Then ayun. Then sa plankton, i-read na siya using itong sa identification and counting na. So, ayan. Medyo hindi ko na alam yung pag-read and identify ng ano kasi it requires yung skills. Yan, talagang pag-aaralan yung pag-identify and counting ng plankton. So, ayan yung sample nung aming pagkuha ng plankton analysis and water quality sa isang lake sa Zambales. So, yun, thank you so much. Thank you for inviting me. Again, I'm Al Dimakibo from the Bureau of Fisheries and Aquatic Resources.
Okay, thank you, Sir Al, for that interesting lecture. I guess the audience is now ready for our speakers to answer questions. Sir Ramjay, Sir Al, ready na po ba kayo? Si
Sige, sir.
Sir Ramjay, nakita ko na sumasagot na ng questions dun sa chat box. Okay, now we proceed to our open forum. To start our open forum, may I call on Ma'am Crystal Joy Malyari to reside as the moderator for our open forum po.
Okay. Can you hear me, Jim?
Yes, ma'am. Loud and clear
Okay.
po.
So, thank you, Sir Ramjay and Sir Al, for an informative and comprehensive talk on water sampling procedure and water quality assessment in inland waters. So with that, allow me to read few questions or clarifications thrown by our Zoom and FB Live participants. So first for Sir Ramjay. So the question is, in the study site assessment form, there is algae under the contaminant box. So the question is, isn't algae actually good because they do photosynthesis in the aquatic environment? So for the follow-up question, is there a specific type of algae that is classified as a contaminant? Sir?
Okay. I think Sir Al can also support me in this answering this question. Pero actually algae, yes, they are source of oxygen. They can be a good source of oxygen for water. However, kasi high amount of algae there's a possibility can cause like eutrophication. Kapag namatay kasi kapag maraming algae sa isang water body, pero kapag namatay yan, nakakadagdag yan sa amin, ang concern kasi namin BOD. So nakakadagdag yan sa biochemical oxygen demand. And also, there's a possibility na nakakapag-block din siya ng sunlight at saka ng dissolved oxygen, pag-penetrate ng dissolved oxygen sa water. So, kapag mataas yung algae content ng water, aasahan mo, pwedeng, yun nga, ma-decrease yung oxygen level dun sa water body na yun. At pwedeng may kamatay ng mga organism doon.
Okay, sir. Dagdagan ko lang yung kay Sir Ramjay.
Sige po.
Tama yung sinabi ni Sir Ramjay. Yung kasing algae, pagkain yan ng mga aquatic organisms, no, ng isda at ng mga invertebrates. So, lagi natin tandaan in moderation lang lagi. Not too much at hindi rin naman yung konti. Sa gabi kasi, nagkakaroon kasi tayo ng hindi na siya nag-undergo ng photosynthesis. Ang ending lahat ng mga organism is nangangailangan pa rin po ng oxygen or dissolved oxygen. So lahat yan mag-aagawan sila, magkakaroon ng competition. So ganun yung mangyayari niyan kapag masyadong marami yung algae. Kasi tinatanong ni ma'am, ano ba yung algae na ano?
Is there a specific type of algae that is classified as a contaminant po?
Yung blue-green.
Oh, okay
Blue-green
po.
algae. Yung blue-green algae, ma'am, siya yung nagkakos ng, ano ba yung tawag sa isda? Yung pag kinain nyo, ano matawag doon? Yung geosmin ng ano doon yung term, parang pag kinain, umok yung pala, sorry, yun yung term, umok.
Okay. Okay po.
Yun ang nagkakos ng ng blue-green algae na yan. Yung nagkakos ng umok sa isda. Sa aquaculture yun.
Okay po. Thank you po. So for the second question, thank you po, sirs. For Sir Ramjay po ulit. So the question is, for collecting and testing water quality samples from the domestic wastewaters, what are the gratuitous permit requirements needed po? And how much is the application for a gratuitous permit will cost? So estimated cost po siguro, sir, or approximate po?
Okay, so actually yun nga, nasanggit ka na. Kasi when we are talking about domestic, tama di ba, yung sa mga household. Actually, kapag household wastewater kasi wala kaming ini-issue permit dyan. I do believe that's under the jurisdiction na ng local government units. And yun nga, dapat nasa septage and sewerage management program nila regarding sa domestic wastewater. So, ang alam ko rin i-require yan, dapat hindi yan directly discharged, dapat nasa mga, yun nga, dapat nasa mga septic tanks and then may collection ng septic tank. So, hindi talaga siya. Pero kapag kung magpapa-test kayo, magkakanda kayo ng test nga nung domestic wastewater, yung best na mag-coordinate kayo sa local government units or kung sarili nyo, kung kayong study, pwede rin naman sa mga third-party laboratories na. Siyempre, kailangan may ang permit nyo kunin na doon sa may-ari ng mga bahay, di ba? Na magko-collect kayo ng sample sa kanila. So, usually, sa amin kasi, depende kasi yan sa laboratory kung magkano yung per test nila. But usually mga 1,000 per parameter, I think, yung iba.
Okay po. Sir Al, would you like to add anything po?
Yung sa ano ba? Yung sa...
For the... okay.
Hindi kasi kami ma'am nagsasample. Kasi kami kasi iba yung mandate namin. We are not actually dun sa yung sa mga sa mga maduduming lugar. Ano. Ang ano kasi namin is sa ano pa rin kami, is feasible siya for aquaculture or for ano, feasible siya for para maglagay ng ano ng isda. Kasi nga we are on the ano po food security sector doon kami sa pag ano doon kami sa plant ng ano na sector ng government. So,
Okay po. Thank you po, sir, for the clarification. So, okay po. Sir, for Sir Ramjay po ulit, so I believe nasagot na din po ito sa ating Zoom chat. But for the sake of our FP Live participants po, so for the third question, what is the best water sampling method to use for collecting water samples from shallow rivers, and any tips po to avoid contaminations of samples during collection and transportation po? So, Sir Ramjay po.
Sorry kasi kala ko kanina live yung answering. Pero ganito, yan, as I answered kasi, if it is just a shallow water body, like mga sapa lang, preferred talaga yung direct method, yung through container na directly from the water body to the sampling container. Pero kasi pero syempre minsan kino-consider din natin yung safety natin in collecting samples. So kung ko-collect ka ng sample through like structures na mga bridges, mga tulay or through boats, edi pwede din yung yung through bucket method na lang. So may intermediate container, dun ka kukuha ng sample and then dun mo rin at mas mainam din yun. And now for contamination, gaya ng nabanggit ko, make sure na properly sealed yung mga container na mga sample containers natin. And yun nga sa amin, in-advise na sa mga tinetest namin, once na na-seal, i-rinse mo yung container prior mo sa ilalagay siya sa mga chest para make sure na hindi siya contaminated. And for fecal coliforms, yun nga, bukod pa doon, kailangan ibalik mo siya sa resealable bag para hindi siya makontaminate ng ibang mga container.
Okay po, thank you po, Sir Ramjay. So we also have a question for Sir Al de Maquibo. So this question is from Mr. Rodrigo Baisa. So it was asked in the Zoom chat box. So the question is there a relationship between ammonia, pH, dissolved oxygen, and temperature? Sir Al, would you like to comment on that po? Sir, nakamute po ata kayo, sir.
Ayun, sorry. Yan, okay na ma'am.
Okay po.
Okay, ang tanong kasi ni sir is the relationship ng ammonia, ng pH, salt, oxygen, and temperature. So yung ammonia kasi, nag-ano yan. So higher temperature, mas higher yung pH. So ang ano niyan is, yung ammonia is toxic ammonia. Sorry. So yung ammonia is mas kailangan niya ng oxygen. So pag nagko-consume yung ammonia ng oxygen, bababa na yun yung oxygen sa tubig. And then maglo-lower din yung pH. Okay po. Yung temperature also, as temperature increases, the dissolved oxygen decreases. So yun po yung relationship ng mga parameters na yan sa water. And sa tubig naman ma'am, pagdating naman sa water quality, very dynamics dynamics talaga ang mga parameters. So wala talaga siya yung kung ano yung reading dito sa isang particular part ng sampling site, same with the other side. So ganun siya ma'am, very dynamic.
Okay, so thank you po for the clarification. So I believe that is all the questions po. Jim, over to you. Thank you.
Okay po. Thank you po, Sir Ramjay, Sir Al, for entertaining the questions of our participants today. Let us all offer a virtual clap for our resource speakers for today, shared their time and knowledge on environmental sampling techniques. I hope everyone got something memorable and noteworthy from our speakers today. Let me just share my screen. Today. Let me just share my screen. To give our appreciation for your passion for science and your devotion, let me just share my screen. To share your knowledge to the people, the Pampanga State Agricultural University, through the College of Appreciation for your passion for science and your devotion, Department of Biology, represented by your knowledge to the people, the Pampanga State Agricultural University, through the College of Appreciation for your passion for science and your devotion, Department of Biology, represented by your knowledge to the people, the Pampanga State Agricultural University, through the College of Appreciation for your passion for science and your devotion, Department of Biology, represented by your knowledge to the people, the Pampanga State Agricultural University, through the College of Appreciation for your passion for science and your devotion. Sorry for that technical problem po. Ayan. Ulit, to give our appreciation for your passion for science and your devotion to share your knowledge to the people, the Pampanga State Agricultural University, through the College of Arts and Sciences, Department of Biology, represented by Dr. Evelyn Totaan, the External and International Affairs Office, represented by Dr. Rafael, the Innovation Center for Agroecology, Climate Change, and Agripreneurship, represented by Dr. Gerald Salas, and the PSAU Biology Society award this certificate. Allow me to read: Pampanga State Agricultural University, PAC Magalang, Pampanga, College of Arts and Sciences, Department of Biology, External and International Affairs Office, Innovation Center for Agroecology, Climate Change, and Agripreneurship, PSAU Biology Society. This certificate of appreciation is hereby awarded to Mr. Ramjay Dizon for his invaluable service as resource speaker during the PSAU Hainayan webinar series, plus PSAU live session with the theme Environmental Sampling Techniques, Soil, Air, and Water. virtually hosted by Pampanga State Agricultural University on August 18, 2021. Given this 18th day of August 2021, signed by the OIC and Vice President for Academic Affairs, Dr. Anita G. David. To award this certificate to our second speaker for today, allow me to again read. Pampanga State Agricultural University, PAC Magalang, Pampanga, College of Arts and Sciences, Department of Biology, External and International Affairs Office, Innovation Center for Agroecology, Climate Change and Agripreneurship, and the PSAU Biology Society. This certificate of appreciation is hereby awarded to Mr. Aldi Makibo for his invaluable service as resource speaker during the PSAU Hainayan webinar series plus PSAU live session with the theme Environmental Sampling Techniques, Soil, Air, and Water, virtually hosted by Pampanga State Agricultural University on August 18, 2021, given this 18th day of August 2021. Signed by the OIC and Vice President for Academic Affairs, Dr. Anita G. David.
Thank you, sir. Thank you. Biosoft. Thank you.
Thank you din po. The link for our post-evaluation form is now available. You may access it in our Zoom chat box and in the comment section of our Facebook viewers. For our students, we would like to remind you once again to please use your personal email instead of your Eskwela accounts in accomplishing our attendance and post-evaluation form. To formally end our webinar, may I ask everyone here in the meeting room to please open our cameras and join us to document this moment virtually through our photo op. Let us all hold our smiles for a few seconds as we go over first slide. I'll also take this chance to invite everyone to join us again on our next PSAU Hainayan webinar series. All information and updates about this will be posted at our official FB page. Just look for PSAU Biology Society in Facebook and follow us. The next session will be on Friday, August 20, 2021. We also remind everyone to answer our post-evaluation form. Again, pakigamit na lang po yung ating personal email accounts para po sa mga PSAU students instead of our eskwela accounts. At this closing instance, thank you for supporting our third day of the PSAU Hainayan second webinar session in collaboration with PSAU Live and the Department of Biology. We hope to see you again soon at our next webinars. Your active participation and support has made this webinar a success. This has been your host, Jimel Peñafiel. Thank you and stay safe, everyone.
Thank you. Thank you, Kabayo. Thank you.
Thank you po, sir. Your active participation and support has made this webinar a success. This has been your host, Jimel Peñafiel. Participation and